Hello everyone. So today we will start with gametogenesis, fertilization and early development. So last video te ami basic concepts of development which is section A সেটা আমি complete করে দিয়েছিলাম. So আজকে আমরা start করছি তাই জন্য gametogenesis দিয়ে. Okay. So first একটা brief intro দিয়ে দিচ্ছি animal development এর cycleটা কিভাবে চলে. First start হয় gametogenesis দিয়ে, which is the formation of gametes. Then it goes on to fertilization, that is the fusion of male and female gametes, formation of the first diploid cell called zygote, and then cleavage starts. Cleavage is a series of cell divisions which gives rise to a multicellular organism. So cleavage এর পর blastula formation হয়, সেটা আমি আগের ভিডিওতে ডিসকাস করেছি. Blastula থেকে gastrula formation হয়, which is the formation of 3 germ layers, and then neurulation, which is the formation of neural tube. Then other organs start developing, which is known as organogenesis, and then it goes back again to gametogenesis. So this is the main cycle. তো দুটো টার্ম ভীষণ ইম্পর্টেন্ট এখানে, একটা হচ্ছে জেস্টেশন আর একটা হচ্ছে পার্চুরেশন. জেস্টেশন হচ্ছে বেসিক্যালি দ্য পিরিয়ড অফ ডেভেলপমেন্ট, লাইক একটা ফার্টিলাইজেশন হওয়ার পর একটা এমব্রায়ো তৈরি হওয়া থেকে ফিটাস স্টেজ অবধি, লাইক টিল ইটস বার্থ যে ডেভেলপমেন্ট পিরিয়ডটা চলে, ইন হিউম্যানস ইট ইজ অলমোস্ট 9 months. সো সেই পিরিয়ডটাকে বলা হচ্ছে জেস্টেশন পিরিয়ড. অ্যান্ড হোয়াট ইজ পার্চুরেশন? পার্চুরেশন ইজ বেসিক্যালি দ্য প্রসেস when the fetus is coming out of the mother's body, or in case of eggs also when it is coming out, which is known as parturition. So next we are going to spermatogenesis and oogenesis. In spermatogenesis it is the production of sperms, and oogenesis is definitely the production of eggs. So there are few basic differences, like it occurs in testis in males, that is already known, and it occurs in ovary in the females, that is also known. So all the stages of this process, the spermatogenesis, occur inside the testis. And in case of oogenesis, all stages do not occur inside the ovary, except the last stage. All the other stages occur inside the ovary. And in case of spermatogenesis, this is a continuous process, that means it goes on after puberty till death. And in case of oogenesis, it occurs in the fetal period only. Like when a girl child is born, when it is in fetus condition in the mother's womb, then only the oogenesis process starts. And the rest of the process, it occurs after the puberty. So this is a basic schematic for the spermatogenesis and oogenesis processes. Like I said, it starts with a diploid cell called spermatogonium. And there are two kinds of spermatogonium, type A and type B. Type A spermatogonium gives rise to only type A spermatogonium, which by mitotic divisions. So it is basically responsible for the increasing the number of spermatogonia in the testis. So this is kind of a stem cell. And type B spermatogonia increases in size and derives nutrition from the neighboring cells and gives rise to a primary spermatogonia. So this primary spermatogonia is also diploid, and from primary spermatogonia, meiotic division starts. Like you can see that secondary spermatogonia is haploid. So after secondary spermatogonia, spermatids, that is this is meiosis 1, this is meiosis 2 basically, and from here we can see that now 4 haploid spermatids are developing from one spermatogonium. So next you can see that they are modified and a tail is added at the end, and then spermatids are called sperms or spermatozoa. So these entire process is divided into 3 phases. This is the multiplication phase. Like I said, the spermatogonia increases in number, gives rise to primary spermatogonia by increasing in size, and this is the growth phase. Because from primary spermatogonia, the secondary spermatogonia is coming by division, and then from growth phase, this is getting into the maturation or differentiation phase. So of course this is a maturation part because the tails are being added to the spermatids. Okay, so From here, as you can see that from one primary spermatogonia, 4 sperms are getting generated. And from one spermatogonia, that means the double number of sperms will get generated. That is 8 sperms will be generated. And in case of oogenesis, similarly, it starts with oogonium, which is a diploid cell, and by multiplication, obviously the same thing is happening. Primary oocytes are getting formed, that is increasing in size, getting nutrition, and then meiotic division starts. So in the first meiosis 1, you can see that secondary oocytes and first polar body being formed. What does that mean? You can see that there is a clear difference in size between secondary oocyte and the first polar body. First polar body is a much smaller cell that is being produced, and the secondary oocyte is a larger cell. So this first polar body will degenerate after some time, and only the secondary oocyte will move into the meiotic 2 division and form the ootate. Here also you can see that one large cell is formed and one small cell, which is called the second polar body, is formed. So this ootate will next move on to maturation and become the ovum. So you can see that from one primary oocyte from one primary oocyte, one ovum is getting generated and 3 polar bodies are formed. One from this here, and the first polar body also divides itself into 2 second polar bodies. So 3 second polar bodies will be formed and 1 ovum. So there is also a difference in the number of final products in these 2 processes. From spermatogenesis, we can say that we will get 8 sperms from one spermatogonium, but from oogenesis we will only get 1 ovum, and the rest 3 cells, that is the polar bodies, they will degenerate. Okay, so this is the spermatogenesis that I was talking about. This entire process can be divided into spermatocytogenesis, which is obviously the formation of spermatocytes. Then spermatidogenesis, which is the formation of spermatids, and spermatiliosis or spermiogenesis, which is the formation of sperms from spermatids, like this step. So if we start with the basic structure of the testis, that is the male sex organ, we will see that this testis is actually connected by a number of ducts, and these ducts are known as epididymis, which move on, and the connection with the urethra is established via another duct which is known as vas deferens. So in this testis, this is in a scrotal sac. So in this testis, the germinal epithelium, that is, it is also formed of a number of tubules called seminiferous tubules, and the tubules are enclosed in an epithelium which is known as germinal epithelium. So this germinal epithelium, if you just zoom out and see the internal structure of it, the transverse section looks like this. And here, as you can see, there are a number of cells attached to the periphery, and also as we move to towards the lumen is the center part of the tubule. As we move towards the lumen, you will see that there are a lot of sperms getting formed here. So these cells, which are the larger yellow colored cells, these cells are known as Sertoli cells or the nurse cells. Basically, they are responsible for providing the nutrition to the spermatogonia, spermatocytes, etc. So starting from spermatogonium, as you can see here, these are spermatogonium. And by mitotic division, like I said, they form the primary spermatocyte, which is quite larger in size than the spermatogonium. And this spermatogonia was derived from mitotic divisions from the primordial germ cell. So the basic arrangement of seminiferous tubules include Sertoli cells and primordial germ cells, and they mainly form the total population. And from primordial germ cells, all the other cells are getting formed. So basically these 2 kinds of cells, they make up the entire population of cells in the seminiferous tubules. So from spermatogonia, as you can see, the primary spermatocyte is getting formed like this. through mitotic division. So it is quite larger in size than the spermatogonium because it has increased its size by deriving nutrition from the Sertoli cells. And from primary spermatocyte, the meiotic division starts. With meiosis 1, the haploid number of chromosomes are getting distributed. Like see, this was diploid, and from here the haploid cells are coming into action. This is the secondary spermatocyte, this stage. So this is smaller in size than the primary spermatocyte. Now secondary spermatocyte will undergo meiotic division 2 and form haploid, 4 haploid cells, that is spermatids. So you can see here that 4 spermatids are getting converted into mature sperm, or it is also known as spermatozoa, and they are getting a tail attached to its end. So this way the mature sperm formation takes place. So here you can see that this is the mature sperm getting formed. This is the spermatid. The spermatid cytoplasm is much larger compared to the sperm. So this cytoplasm is shed off, a tail is added, and it is much more slender, and so that it helps in the motility of the sperms. These changes are being made from spermatids to spermatozoa. So in this way the maturation occurs. Now if you can see here the 3 stages. The first is the spermatocyte formation. Up to this stage is known as spermatocytogenesis till secondary spermatocyte formation. From here to spermatid formation, it is known as spermatidogenesis. And the last stage, which is the mature sperm formation, this is known as spermateliosis or spermiogenesis. Next, moving on to the structure of a sperm. So as you can see here, this is known as, this is divided into 2 major parts, the head and tail. And this you can call it a neck portion, because here the mitochondria is aggregated over here, and it is known as a midpiece. This tail is basically a flagella. Like flagella helps in movement of many organisms, the sperm also has a flagella and it helps in its motility. This mitochondrial midpiece is mainly responsible for providing the energy so that the flagella can move and the motility is enhanced. Next we have the nucleus. So you can see that major part of the cytoplasm is cut off and only the head contains the nucleus for a larger part of the head. There is a cap which is known as the acrosome, and the centriole lies here. So this is the basic structure of a sperm. Now moving on to the hormonal control of spermatogenesis. So hormonal control, that we already know that hypothalamus is the main center from where the master gland, the anterior pituitary, gets signals, and these signals are nothing but different kinds of hormones, and from here the anterior pituitary secretes certain hormones which move on to act on certain target organs. So here we are concerned with the hypothalamus pituitary testicular axis, which is the major hormonal pathway of spermatogenesis. So from hypothalamus, the gonadotropin releasing hormone is secreted. So by gonadotropin, you can understand that it has something to do with sex organs, gonads, and it triggers mainly the anterior pituitary to secrete the gonadotropins, which is FSH and LH. So FSH is follicle stimulating hormone and LH is luteinizing hormone, also known as ICSH, interstitial cell stimulating hormone. And these two hormones, they have different functions. Obviously, FSH, that is the follicle stimulating hormone, goes on to the testes to act on the Sertoli cells, and from the Sertoli cells they stimulate the spermatogenesis. Inhibin is secreted and ABP is secreted. ABP is androgen binding protein. Androgen is basically the male sex hormones are known as androgens. Obviously the major androgen being testosterone. So that testosterone is secreted by the Leydig cells or the interstitial cells of Leydig. Therefore the name ICSH, interstitial cell stimulating hormone, is given to LH, because LH triggers the Leydig cells to secrete the testosterone. This testosterone is responsible for stimulating the Sertoli cells so that they trigger the spermatogenesis and they enhance the process, they increase the number of sperms inside the testis. FSH also acts on spermatogonia and so that it divides more rapidly and it helps in the multiplication of spermatogonia and thereby formation of sperms. So inhibin is another hormone which is responsible for creating a negative feedback loop. As you can see that inhibin being released by the Sertoli cells, they go on to anterior pituitary as well as hypothalamus and stop the secretion and try to inhibit the secretion of GnRH from hypothalamus and FSH from anterior pituitary. So this is a negative feedback loop. When the spermatogenesis is stimulated enough and it needs to be stopped, the hormone action needs to be stopped, otherwise it will go on and on. So some regulatory mechanism must be there to stop this hormone secretion. Therefore, inhibin is secreted by the Sertoli cells to control this process. And next is the testosterone. Testosterone also has an inhibitory effect by negative feedback loop. It goes on to inhibit LH secretion from anterior pituitary and it goes on to inhibit the GnRH secretion from hypothalamus. So as you can already see that LH is the main hormone which triggers the secretion of testosterone here. So when testosterone concentration or the amount of testosterone has increased to a certain level, it has reached a threshold, then no more testosterone is required in the body. So it goes on and creates a negative feedback loop, stopping its own secretion. So this testosterone is then responsible for various functions which we can see here, that mainly the testosterone is known as a sex hormone responsible for the sexual functions of men, and but we can see that testosterone has some other functions as well, like with heart health, it lowers the cholesterol levels, improve the cardiac capacity. In brain, it has been showing improved results in cognition and improved focus, memory and mental health functions. As well as in metabolism, it increases lipid oxidation, normalizes glucose utilization and lowers the risk of diabetes type 2. And also in blood circulation, it is helping in the production of RBCs. It can help counteract the effects of anemia. Of course, when anemia, red blood cell formation is helped by testosterone, then obviously anemia will be decreased. So testosterone and central nervous system, individuals diagnosed with low T often report feelings of depression and treatment. So obviously it is also helping in curing depression. In some level, it can improve the overall mood of a person, and of course in sexual functions it is responsible for the sexual arousal in men, and obviously for decreased libido, testosterone is very important. Next, moving on to ovulation or oogenesis. So from ovulation, as you can see, this is the internal structure of an ovary, and like I already said that this primordial germ cells are here also. In spermatogenesis, primordial germ cells were the first cell to begin with, and in oogenesis the same primordial germ cells are there. So it will form the oogonium by differentiation, since germ cells are stem cells basically. So by differentiation, they are forming the diploid oogonium. Oogonium can divide by mitosis and create more and more oogonia. And from oogonia, the primary oocyte is formed, which is a much larger formation of the larger in size than the oogonia, and therefore the primary oocyte is moving on with the meiotic division, and it creates the secondary oocyte and the first polar body, which is a smaller cell. It goes on to degenerate later, but after it forming two other polar bodies in the next second meiotic division. Secondary oocyte forms another larger cell and another smaller cell which is known as the second polar body. This cell, larger cell, is known as the ootid, which is definitely haploid. In secondary oocyte, this haploid cells are being formed from here to here, and you can see that this ootid is getting converted into an ovum. So there are a few changes by which the ootid gets converted into an ovum. And here you can see that from these are primordial follicles, and the one which is a little bit larger in size, this is known as a primary follicle. The center part of the follicle, you can see there is a smaller structure which is known as the ovum. So this is the ovum, primary ovum, and this ovum is surrounded by a number of cells which only some cells of the oogonia become primary oocyte, and the rest of the cells help in the follicle formation, which is nothing but the outer external covering of the ovum. So from here, as you can see, that the follicle size is increasing, and this follicle is getting larger and larger stage by stage. So these are developing follicles, and ultimately the secondary ovum is formed, and this is the highest level of maturation of the follicle, and this follicle is known as a Graafian follicle. So after the Graafian follicle is formed, it fuses with the ovarian wall, and the follicle ruptures and liberates the ovum in the fallopian tube, and then it goes here. So from the fallopian tube it goes into the uterus. So this, after this rupture has happened, this is the early corpus luteum. The Graafian follicle is now known as a corpus luteum after releasing the ovum. So this starts degenerating and it becomes the corpus albicans. So this is the entire cycle which is completed in the ovary. So according to that, the phases are named as follicular phase, ovulation phase and luteal phase. So up to the follicle formation, up to Graafian follicle formation, it is known as follicular phase. The moment it liberates the ovum, this is known as ovulation phase, and from here till corpus albicans formation, the corpus luteum is the primary structure here, so this is known as the luteal phase. Okay. So now this is very interesting that the oogenesis, like I already said, it starts from the fetus stage. Like whenever a girl child is getting formed inside a mother's body, the oogenesis has already started in the fetal condition. So before birth, as you can see that from germinal epithelium, oogonium is getting formed by mitotic divisions. And this diploid cell is stuck at prophase 1. Mitotic divisions are going on, and primary oocytes are getting formed, and it is arrested in prophase 1 until puberty. So at the time of birth, 2 million primary oocytes will be formed, and in the fetus or in the girl child that is being born. So you can see that the oogenesis has already started before birth, and at birth only the female child will have a number of primary oocytes. So this will be arrested at the prophase stage of meiosis 1. Next, moving on to the completion of meiosis 1. So when it reaches the puberty, so when the girl child, she reaches the puberty stage, then the meiosis 1 moves into completion. So here this FSH hormone triggers this completion of meiosis 1, and of course the first polar body is getting formed and the secondary oocyte is formed, which is the larger cell. So this secondary oocyte then is arrested at metaphase 2. So here you can see that once a month this prophase 1 moves to metaphase 2 during ovulation. So after puberty, every month this secondary oocyte will be formed. And from here you can see that at puberty FSH starts being produced, so the secondary oocyte will move on to the after getting released from the ovary, when it reaches the uterus, if the sperm entry has occurred, then it will be fertilized by the sperm. And this sperm entry will trigger the completion of meiosis 2, that is the fertilized egg produce or the ovum production, and the second polar body will be released. So here you can see that the total oogenesis process is occurring at 3 different stages. So in case when the fertilization has not occurred, then the secondary oocyte gets released every month by menstrual cycle, and here when fertilization has occurred, then the secondary oocyte is getting converted to the ovum or the fertilized egg. So here also you can see the same thing, that this first polar body is degenerated, it never becomes an egg, and this secondary oocyte which was arrested at metaphase of meiosis 2, this after ovulation, when the sperm entry has happened, then only it moves on to become the final stage of like the ovum formation has been complete, because meiosis 2 was not complete and it was arrested in metaphase stage. Anaphase comes after sperm entry has occurred. Sperm entry basically triggers this process.